The story of Jewish ancestry begins deep in the ancient Near East, long before written history, when the first humans settled the Levant. Genetic evidence traces the earliest roots of future Jewish populations back over 12,000 years, to the Epipaleolithic Natufian communities. These hunter-gatherers carried a unique blend of basal Eurasian DNA, a branch that split off early from other Eurasian groups. Their genetic signature, especially lineages within E1b1b and early forms of haplogroup J, became the foundation for all later Levantine peoples, including those who would eventually form the ancestral population of the Jewish people. As the region transitioned into the Neolithic era, agriculture spread across the Levant. These early farmers kept much of the Natufian genetic base, but they also absorbed new ancestry from neighboring populations. This mixture formed the demographic platform from which Semitic-speaking groups, including the Proto-Canaanites and later the ancient Israelites, emerged. By the Bronze Age, between 2500 BCE and 1200 BCE, Canaanite societies flourished. Ancient DNA recovered from sites such as Megiddo, Sidon, and regions across modern Israel and Lebanon shows that these people carried a remarkably stable genetic profile. A blend of local Neolithic Levantines and incoming populations from the Zagros Mountains and the Caucasus. This genetic mixture became the backbone of Jewish ancestry. Their Y chromosome patterns, especially J1, P58, J2, M172, and E1b1b, remain deeply embedded in Jewish populations today. Autosomal DNA studies confirm the same story. Modern Jews share a strong genetic link to these Bronze Age Canaanite communities. When Israelite culture emerged during the Iron Age, around 1200 BCE, it did not arise from outsiders replacing local people. Archaeology and genetics both reveal that the Israelites were essentially Canaanites who developed a new cultural identity, shaped by different religious ideas and social practices, not by a new population. A small amount of gene flow from neighboring groups like Arameans and Phoenicians occurred, but it did not change the core genetic structure. The Assyrian and Babylonian exiles brought new influences to Jewish communities living in Mesopotamia. Iraqi and Iranian Jews still carry traces of these ancient interactions, though their dominant ancestry remains firmly Levantine. Meanwhile, many Judeans never left the region, helping preserve continuous genetic links to the ancient homeland. During the Persian and later Hellenistic periods, Jewish communities spread widely across the empire. Contacts with Greek and Anatolian populations introduced limited new lineages, some seen today in haplogroups such as G2A, T, and R1b. The Roman era expanded this diversity further as Jewish communities settled across Europe and North Africa. The Jews of the Iberian Peninsula, who later became known as Sephardim, developed a distinctive mix, roughly two-thirds Middle Eastern ancestry blended with Mediterranean and Iberian DNA. Their paternal lines remain strongly Levantine, while maternal lines show greater European diversity. Mizrahi Jewish communities in the Middle East and North Africa, however, preserved the closest genetic continuity to ancient Levantine populations. Groups from Iraq, Iran, Syria, Yemen, and North Africa show exceptionally high frequencies of J1, J2, and E1b1b, reflecting thousands of years of continuous presence in or near the original homeland. Yemenite Jews stand out as a case of extreme genetic isolation. Despite living far from the Levant, they retain strong Middle Eastern ancestry with very little Arab admixture. Ashkenazi Jews, today the largest branch of world Jewry, carry a unique genetic signature shaped by migration, admixture, and historical bottlenecks. Their ancestry is roughly half Middle Eastern and half Southern European, reflecting early migrations through Italy before they moved into Central and Eastern Europe. A severe population bottleneck in the Middle Ages reduced genetic diversity, amplifying certain rare variants and contributing to the founder effect genetic disorders seen today. Despite this bottleneck, Ashkenazi paternal lineages remain heavily Middle Eastern, while maternal lines show a strong European component, evidence of ancient intermarriage and conversion during the formation of early Ashkenazi communities. One of the most striking genetic patterns is found among the Kohanim, the traditional priestly lineage. Across Jewish populations, Ashkenazi, Sephardi, and Mizrahi, men who identify as Kohanim often share a distinctive Y chromosome pattern known as the Cohen modal haplotype, rooted in haplogroup J1, p58. Genetic dating places their common ancestor around 3,000 years ago, aligning with the biblical timeline for the early Israelite priesthood. Some Jewish communities, such as Beta Israel in Ethiopia and the Bene Israel of India, preserve Jewish cultural and religious traditions while having genetic profiles that reflect their local populations. These groups highlight an important point: Jewish identity has always been both genetic and cultural, shaped by shared traditions as well as shared ancestry. Today, in modern Israel, marriages across different diaspora backgrounds are blending once separate lineages. This is creating a more unified genetic landscape, one that brings together threads from every branch of the Jewish world. In the end, genetic research reveals a clear picture. Jewish populations around the world share deep roots in the ancient Levant, shaped over thousands of years by migration, isolation, adaptation, and cultural continuity. Their DNA records not just biological inheritance, but the entire arc of their history. Thanks for watching. Please like and subscribe.